All right. Thanks very much for having me tonight. It's a pleasure to talk to you about the PhD project I'm working on under the supervision of Tristan Barrett and Ferdy Gallagher here at the department, dedicated to hyperpolarized MR of prostate cancer. And tonight we're going to talk about the intersection of biology versus imaging. So I know that the audience here is unlikely to be of a lot of prostate cancer experts, so I'll try to be very clinically focused in my presentation. So prostate cancer is incredibly common. The second commonest, the fifth deadliest cancer, and the incidence is actually going to continue to rise and will double by 2040. So we actually have to do something about that. So in terms of all the new cases, that's an oversimplification, but this will help you put the whole thing in context. Around 50% are locally advanced or metastatic disease, so we treat those patients without any questions. But 50% of those have organ-confined disease, and here we have different options. Well, there's no clear consensus across the whole world, but approximately 20% of those patients would require immediate treatment because they'll have large Gleason 7 tumors with high percentage Gleason pattern 4, and that's the metric we're going to go back into this presentation quite a lot, or they have Gleason score 7 and above tumors with high component, and 30% of those patients are actually eligible for active surveillance. So we're trying to defer treatment and preserve the quality of life for as long as possible. And those are patients with Gleason 6 tumors or Gleason 7 with low percentage GP4 component. These tumors are unlikely to progress or kill the patient. But actually, 50% of those patients on active surveillance do progress over 5 years. So actually, we're doing something wrong either when selecting those patients at baseline with inherently progressive tumors, or we're not good enough at picking up progressions early on to switch to treatment more effectively. And the clinical challenge that we're going to try to tackle today is differentiating indolent versus clinically significant prostate cancer, what we don't treat and what we treat now. And we need to be better here because if we're using pre-biopsy multiparametric MR as a gold standard, so when we're trying to detect clinically significant prostate cancer, we are very good at ruling it out with 90% negative predictive value. But when we're seeing a lesion, actually, 35% we're good at telling that this is clinically significant, but not indolent. So we need to be better here. And well, we try to harness not just metabolic or functional metrics available as part of multiparametric MRI, but something different. And metabolism and deregulated cellular metabolism is one of the hallmarks of cancer that we all know. And hyperpolarized MR in the first-in-man study, in fact, was done in prostate cancer, whereby you could see increased lactate production in histopathologically confirmed areas of prostate cancer compared to the benign tissue. That was the first-in-man study in UCSF back in 2013. Since then, the same group has shown that you can also image metastatic prostate cancer pretty well with this technique. Then a group from MSKCC in New York showed that actually as your grade group increases, you're seeing higher lactate production in those tumors. And then the same group from UCSF showed that following androgen deprivation therapy and immunotherapy, you do see a decrease in lactate production in those tumors. So you could potentially use the technique to first risk stratify those tumors when detecting them and evaluating treatment response in that setting. However, we do still have lots of questions that have been unanswered and that are critical for clinical translation of this technology. So two questions we're going to tackle today as part of this presentations are, so how does lactate labeling in hyperpolarized MR relate to standard of care biomarkers of the disease, i.e. multiparametric MRI, cellularity and perfusion, and histopathology? So does this actually add any value to what we have in the clinic? And then how can we explain this differential lactate labeling in those tumors of different aggressiveness? There have been attempts to do that, but none of them have been really comprehensive. So we'll try to see what we're doing. Is it just, again, cellularity or perfusion which differs in those tumors, or is it actually metabolism? So with that in mind, we did set up the hyperpolarized MR facilities here in Cambridge for prostate cancer. And this was the first study that I'm now going to talk about. That's the first study in Europe to do hyperpolarized MR in prostate cancer patients. So we had pre-biopsy and PMRI in patients. We identified 10 of those with 15 really visible lesions. 2 of those were low grade, the majority of those were intermediate grade disease, and 2 were what we would call clinically significant cancer. We then performed hyperpolarized MR before radical prostatectomy, where we took the samples out and measured percentage Gleason pattern 4 as a more refined metric of aggressiveness of those tumors due to the overrepresentation of this disease. So if we took grade, there would only be 3 grades, and the representation will be skewed towards this. So this was a more clinically relevant metric we chose. So here is a very good example of what we showed. You can see a patient, Pullman prostatectomy specimen with a lesion in the left side of the peripheral zone. It's 50% GP4, grade 3 disease. You would treat this immediately. And on the contralateral side, in the transition zone, you would see a grade 1, 3+3 lesion with 0 GP4 that was not detected on standard of care MR, on ADC or DCE, but we could pick it up on hyperpolarized MR. So the first thing here was, okay, well, fine. Let's try and see whether our lactate labeling had any relationship with the standard of care markers that we've discussed. And the first one was that we did see the significant positive correlation between lactate SNR and percentage GP pattern 4, which was good. But then we also saw a negative correlation with ADC. So, okay, well, if it's just cellularity, then maybe we're not adding much to standard of care. And we then saw no relationship with K-trans. Well, we have a low sample size, you could argue, but still, there was a link with ADC, but nothing with K-trans. So first of all, just in this simple slide, we could identify MR occult disease that was not present in the standard of care, and that's particularly relevant in patients who are considering focal therapy. You wouldn't do focal therapy in this guy, in whom you see disease on both sides of the gland. Then we could image percentage GP pattern 4 non-invasively, which is a clinically useful marker we'll discuss in future. And then there was no effect of perfusion, at least in this sample size, on what we were observing. But let's go dive deeper into the biology, because what I've shown to you has roughly been known before. So if we look at the benign prostate is a very interesting organ, because unlike many other cells in the body, it's in fact not oxidative, but glycolytic. The point is, the function of the prostate is to export citrate in the seminal fluid. And in order to do that, you need to truncate your TCA cycle, and therefore your mitochondrial metabolism is deficient. So to compensate for that, you would rely on ATP production from pyruvate to lactate conversion. But when these epithelial cells become malignant, at least in the early stage of prostate cancer, you see that there's a loss of inhibiting impact of zinc on the TCA cycle. The TCA cycle is restored, and it is more energetically efficient. These cells, in fact, become more oxidative rather than glycolytic. So this is the reaction we would think will be imaging in the cohort that I'm describing. And as the tumors progress towards a more aggressive phenotype, the mutations accumulate, the true hypoxia within the environment accumulates, and therefore these tumors become truly glycolytic that you can then pick up, for example, on FDG PET. And you know that these lesions you cannot pick up because they are largely oxidative during their phenotype. So how can we harness that in terms of our own work? So first of all, we identified the link with cellularity, but ADC is crude cellularity. It's epithelial cells and stromal cells. We can't tell the difference. An important thing is that both epithelial cells and stromal cells are metabolically active in prostate cancer. They both produce lactate to a certain extent. So what's their differential contribution to what we're seeing? So we had the prostates out, we classified the tissues to epithelium and stroma, and then we first measured the relationship between the epithelial cell number and lactate SNR and the stromal cell number and lactate SNR. And we showed that actually it was the epithelial cells that had the more prominent relationship with lactate, which made us think, okay, that perhaps what we're seeing on imaging is the epithelial compartment. But we needed some additional explanation to that. And what could be the explanation and the mechanism supporting this is the expression of MCT1. That's the monocarboxylate importer, which has a higher Km for pyruvate than for lactate. And therefore, if it's overexpressed on the cells, it's more likely that pyruvate that you inject into the tissue of interest will be uptaken by those cells. And in fact, we stained the tissues for MCT1 and MCT4, which is another component that exports lactate from outside the cell. And we saw on this graph that MCT1 is expressed significantly, all of it's almost on the epithelium, and you can see it here clearly on this sort of slide. So first of all, you got a correlation between epithelial cells and lactate SNR, and then you have all your MCT1 in this cellular compartment, from which we think the hyperpolarized MR does not just measure cellularity, but epithelial cell density. And that is really important to what we're measuring. And that's what adds value to standard of care. And actually, okay, let's go next. Well, what's actually driving the clinical lactate labeling? And what is more important, the total LDH expression or the isoenzyme pattern? You may argue that in this tissue, in a steady state where there's no clear flux one way or another, it's not the isoenzyme pattern that matters when you inject a lot of pyruvate. It's the total enzyme which will swing the reaction in one direction from pyruvate to lactate. So what we did was we did some spatial transcriptomics with LDHA and LDHB, and we could measure them separately or we could combine them. And what we saw that actually very strong correlation between total epithelial LDH and lactate labeling, but nothing really meaningful between LDHA and LDHB. So that's really the total enzyme that matters. And previously it was only LDHA that people looked mostly at. And then in terms of, in addition to that, we know that as soon as cells start producing lactate, they need to get rid of it because it's cytotoxic, and they do get rid of it through MCT4. And in fact, you would expect that in cells that are producing lactate, MCT4 would become more expressed, and normally it's all in the stroma. But what if, when the cells become aggressive, they start overexpressing it, and we visualize it in the form of epithelial to stroma MCT4 ratio, which again showed a high positive correlation with lactate SNR. So cells that have a lot of LDH produce a lot of lactate, and the same cells need to pump it out through the migration of MCT4 on them. And that's sort of one of the potential explanations for what we see. But then, okay, we had this correlation with lactate SNR and high GP4 and cellularity, but is it all just noise, or can there be any metabolic explanation to that? So first we measured MCT1, MCT4 in those cells, and LDHA and LDHB, but also PDHA1, so all the key metabolic enzymes that can be playing part in the reaction we're imaging. So what we found was, in terms of MCT1, there was no difference between tumors with low and a high percentage GP4 component. In terms of MCT4, there was a significant increase in the epithelial compartment, but not necessarily in the stromal compartment, although sample size is pretty low. LDH went up, LDHB went down, PDHA1 went down, suggesting the possible shift from the excidative phenotype to glycolytic, as we've discussed. And the epithelium to stromal ratio goes up, the total epithelial LDH goes up, and the LDHA to PDHA1 ratio is a sort of a surrogate of glycolytic versus excidative metabolism, also goes up in those high GP4 tumors. So those tumors do show a shift towards glycolysis that could explain those hyperpolarized MR findings. But then we went even deeper. Okay, we're looking at the level of the whole glands, which is the resolution we can sample in clinic with using hyperpolarized MR. But what happens at the individual glands? We had GP3, GP4, GP5, and as you go, for example, with MCT1 component, you would see the increase towards the higher grade disease. In terms of epithelium to stroma ratio, you would see it's all in the stroma here, but somehow in GP5, MCT4 is all in the epithelial compartment. And then the total epithelial LDH, again, goes up towards the high-grade disease, as well as LDHA, PDHA1 ratio. So these glands do harbor a distinct metabolic phenotype, suggesting progression towards glycolysis. Well, and finally, okay, we see the increase in LDHA. Can we get another surrogate marker to kind of validate that, if you like? And we know that it's a transcription target of HIF1 alpha. So then we looked at the epithelial LDHA and the nuclear HIF1 alpha where it exhibits its activity. And we see again that in those GP5 glands, HIF1 alpha in the nucleus is being translocated there, which could potentially explain at least the increase in the expression of this specific gene we're looking at. And do these findings hold in larger data sets? Well, yes, they do. We took the TCGA data and showed that roughly the same trends we're seeing, they do hold there. So in conclusions, the two questions we asked, so how does that relate to standards of care? For proton MR, we detect MR occult disease, and that's important for active surveillance enrollment. Patients with more tumors will require more stringent surveillance, and they would not qualify for focal therapy. We do correlate this with ADC, but we are being specific to the metabolism in the epithelial cell compartment, and that's more than just crude cellularity. And in terms of histopathology, we can non-invasively profile GP4 disease at the whole level lesion, so we don't have any sampling error with the biopsy. And finally, total epithelial LDH, epithelium to stroma MCT4, and that LDHA to PDHA1 ratio are all tissue-based markers that reflect those changes we're seeing in hyperpolarized MR, bringing us a bit closer to understanding the biology versus the clinical meaning of that technology. Thanks very much for your attention. Any questions we'll take now or later.